Hi everyone, this is your biology class. I'm your teacher Seema Treja. Student, in previous video we discussed the concept of vector which help us to transfer the desired gene into the host cell. In this video we are going to discuss how to make the host cell competent that it easily take the recombinant DNA. Two types of cells exist in nature: prokaryotic cell and eukaryotic cell. If we use the prokaryotic host cell, most commonly used cell is the E. coli. In case of eukaryote, host cell may be animal cell, may be plant cell, or fungal cell. Most commonly used cell as a host in case of eukaryote is the yeast, which is a type of fungal cell. किस तरह से हम होस्ट सेल को कॉम्पिटेंट बनाते हैं? First step what we do, we remove the cell wall. It means we use the protoplast. For this, we remove the cell wall if it is present. Second step, we treat the cell with divalent calcium ion. As we all know, plasma membrane is hydrophobic due to presence of bilipid layer. And DNA is hydrophilic and negatively charged. So this hydrophilic DNA cannot cross through the plasma membrane. So we treat the cell with divalent calcium ion. By doing this, permeability of plasma membrane increases. Third step we done here, we treat the or incubate the cell with recombinant DNA on ice. Then pass this in an environment where temperature is 42 degrees Celsius. What we called it as heat shock. After giving heat shock of 42 degrees Celsius, we again pass this on ice. This enables the bacteria or host cells. This enables the host cell to take up the recombinant DNA. So in this way we make the host cell competent. First we remove the cell wall, then we treat this cell with the divalent calcium ion, so the permeability of plasma membrane increases. Then we incubate the cell with recombinant DNA on ice and then pass this cell with recombinant DNA into the environment where temperature is 42 degrees Celsius. In this way, we give the cell heat shock treatment. After that, we again shift this on the ice. In this way, we make the cell competent, so it enables the host cell to take up the recombinant DNA. One more thing we are discussing here, some vectorless gene transfer method. These are the methods in which vectors is not used to transfer the desired gene into the host cell. One such method is the chemical method. क्या करते हैं chemical method में? We use the protoplast, that is cell without cell wall. We put this protoplast along with desired gene into a medium containing PEG, that is polyethylene glycol. In the medium of polyethylene glycol, gene directly enter into the host cell. Vector is not required here to transfer the desired gene into the host cell. So it is a vectorless gene transfer method. Second method, which is again vectorless, is known as the gene gun. Method. We also called it as shotgun method. We also called it as ballistic method. What we done in this method, we used micro particles of gold or tungsten coated with DNA, and cells are bombarded with high velocity microparticles of gold or tungsten coated with DNA. In this method, we prepare the microparticles of gold or tungsten coated with DNA, and we bombard over the cell so the desired gene or DNA enter inside the host cell. Third method is the micro injection. क्या करते हैं माइक्रो इंजेक्शन में? Recombinant DNA is directly injected into the nucleus of animal cell. This method is used to transfer the desired DNA or recombinant DNA into the animal cell. Recombinant DNA is directly injected into nucleus of the animal cell. In the form of animal cell, mostly use the egg cell. The nucleus is eccentric in case of egg cell, and we directly inject the recombinant DNA into the nucleus of the animal cell. This method is known as the microinjection. One more method we discuss here, that is electroporation. Electroporation. क्या करते हैं इलेक्ट्रोपोरेशन में? We take cell in a medium and pass into high voltage electric field for a very short period. We pass the host cell into a field where desired gene, that is recombinant DNA, is present, and we create a high voltage electric field. For a very short time. When we create the high voltage electric field for a very short time, it creates a hole into the host cell. It is a temporary small pore develops in the membrane of the host cell. At that time, this recombinant DNA through this hole enter inside the host cell. So vector is not required in this method also. In case of electroporation, what we did, we passed the cell into the medium where recombinant DNA is present, and in this medium we create the high voltage electric field for a very short time. When we create the high voltage electric field, a small pore develops into the membrane of the host cell, which is temporary. And at that time, this recombinant DNA through this hole enter inside the host cell. When recombinant DNA enter inside the host cell, this hole is again blocked by cell membrane itself. So these are the methods in which vector is not used. These are the method where directly pass the desired gene into the host cell without using the vector. This is all about in this video. Thank you so much.